All right. So I think all that, I got it again. Still don't think I can fill it. All right. So then, if we look at the next one, is telophase 1. Oh, there the tetrads being pulled apart. And now, telophase 1. We have the actual homologs reaching the pole. All right. All that spindle fiber in there starts to disappear. The chromosomes, though, stay condensed because we got to go through another division. And normally, what happens in telophase? Normally, in mitosis, the nuclear envelope reappears, right? But in meiosis, we're not going to do that. And the reason being, we now have two cells once cytokinesis forms. Okay, cleavage is finished. We have less chromosomes. We had 6 before, now we only have 3 in each. However, we still have replicates there, okay? And we can't have those. That would mess up things, all right? Because that would add DNA to, four strands of DNA per pair when they are during fertilization than just 2, right? So we've got to go through another division to get rid of those extra DNA molecules. And that's meiosis 2. And in meiosis 2, it's just mitosis, all right? So I'm just not going to, I drew it again while we were on meiosis 1. So just we'll make sure. Cytoplasm divides between each of the daughter cells during cytokinesis. Cleavage in the animal cells pinches the 2 new daughter cells apart. And so we're finished with meiosis 1. We have 2 daughter cells. They are haploid, which is what we wanted. But the chromosomes are still duplicated or replicated. Okay? So another division is required. What is not going to happen between meiosis 1 and meiosis 2? If we have DNA already replicated, what are we not going to have happen?
More DNA replicated.
That's right. So we're not going to have interphase, right? We don't want more. We're trying to get rid of it. So we got to have another division. We're not going to go through interphase because we don't want more DNA. All right? We want to get to reduce it. Okay? So there's no interphase between meiosis 1 and meiosis 2. So we started with this diploid cell. Okay? It has, I'm going to just make 4. Okay? It has four chromosomes. It went through replication during interphase, so it has these replicates of itself. And after meiosis 1, all right, we have 2 haploid, right, because now we have 2 in each versus 4, but those are still replicated. So we still have to reduce the number of DNA molecules. Right? And that is what meiosis II does. Okay. So the meiosis II, I'm not going to spend a whole lot of time on because, like I said, it's the same. It's exactly like mitosis. There's meiosis telophase 1. All right. So there's no interphase between the divisions of meiosis I and meiosis II. Therefore, DNA is not duplicated or replicated. So, prophase 2. Spindle fibers form from centrioles. They connect to the chromosomes. They connect to the other spindles in the opposite pole. All right. Okay. That again, same thing that happens in prophase mitosis. The chromosomes are already condensed, and the nuclear envelope's already disappeared. It's already gone. Right? So it doesn't have to do that. Okay. And so you got now, in both of those two that I've got drawn there after meiosis I, you've got the chromosomes starting to be, the centrioles starting to move. And then in metaphase 2, the chromosomes line up, single file, on the metaphase plate, just like in mitosis. All right. Same thing. So there they are, lined up. Okay. And then anaphase 2, the chromatids then are pulled apart at the centromere. Okay. The cell elongates, and those chromosomes then now move toward the poles, the resulting chromosomes. All right. So here we got those sister chromatids being pulled apart. They're moving toward the poles. They should be a little more elongate than that, but that's okay. And then cleavage starts, cytokinesis occurs, starts with telophase 2. And now, once the chromosomes have reached the poles, the nuclear envelope will reform, okay? Just like in telophase of mitosis, all right? Spindle fibers, they all disappear. And then once the two have pinched off, meiosis II has ended. So now, after meiosis II, all right, we started with 4, right? We reduced it to 2, but they still had 4 DNA molecules. So then we went to another division. Now we have 4. And they are all haploid, and they all have 1 DNA molecule, so we're all happy. Okay? So, from a meiotic division, you start with a diploid parent cell, and you end up with 4 haploid gametes. All right. Any questions about meiosis? And this only occurs in the testes and the ovaries. Right? All the rest of the cells will just do mitosis. End result, 4 haploid gametes. right, so if this is an ovary, all right, so it's easier with, let's start with this. All right, you have in the male reproductive organ, and that's Mars, okay, so that's a symbol for male. You have a parent cell who undergoes 2 meiotic divisions. The result are 4 sperm, okay? Meiosis occurs till really old, especially for you all, really old, okay? 60s, 70s, all right? Men still can produce sperm, viable sperm, okay? So this occurs. Meiosis in the testes also doesn't start till sexual maturity, all right? So that's textbook, right? 4 haploid gametes. In the women, so in the ovary, and we're going to make this Venus now instead of Mars, okay? You have a diploid parent cell. It undergoes meiosis 1, meiosis 2. But for each meiotic division, let's get rid of the tail. Only really one becomes viable. All right. So 1 ovum, 1 egg. These others actually get absorbed by the ovary, by the egg, ovum, right? These are called polar bodies. And if you, now I don't know how this happens. I mean, I can sort of, if you're a female, if you look at a cheek cell, for instance, underneath the microscope, you will see on the nucleus these structures, thickened places, and they are the polar bodies. All right? I don't know how they get there. Magic. I don't know. They wind up on the nucleus. So you can look at a cell and see if it's male or female, because you can actually see those little polar bodies. They're thickened enough where you can tell that there's something there on the nucleus of the female cell. All right. But these are all absorbed by the egg. It eats them. Right. Adds more nutrients for the egg. All right. Meiosis occurs actually in development, in some cases before birth, all right, in the ovary. And it occurs throughout a certain set amount of time, right? At some point in time, the ovaries stop the meiotic process, right? So there is only a finite time where those eggs are produced. Once they're finished, there aren't any more, okay? Then, females go through what is called menopause, right? That's the time when the ovary is stopped producing eggs. All right. Now, I'm sorry. Production is even earlier than menopause. Okay. Production probably stops at the peak of your physical life, all right, in females. So 20s and 30s maybe, all right. So there's only a certain amount of eggs. Once the female reaches maturity, they start releasing eggs every month, right? And, you know, they just keep on releasing until finally you hit the point where there's no eggs left, right? And so that's when menopause starts, right? And with that, you know, changes in hormones and all that kind of stuff, all right? Because the release of eggs spurred different types of hormones that are not as plentiful as they were when you were releasing eggs, okay? So it changes things, and so, you know, females, the meiotic divisions are a little bit different than in males as far as that, you know, timing and all that kind of stuff. All right, any questions about that? So, hopefully, it's turned back on. So the last part, the last little bit of chapter 8 deals with talking about genetic variation, all right? How you can get a little bit of differences in the traits, expressions of traits, by the way things are sort of way they go, I guess, during certain events of the cell cycle. So one thing that can change the expression of a trait, for instance, whether you can roll a tongue or where you can't, okay, can be independent orientation. All right, this is where during metaphase 1, all right, when the homologs are pushed to the center of the cell, I guess that. This out of the way. All right, so here's your metaphase 1, and here are your homologs lined up in the center of the cell. Maybe they line up like this, okay? Maybe like this. All right. So how they're pushed into the center of the cell can depend on which of these goes to which of the daughter cells, right? So if tongue rolling was on this particular homolog pair, and this one could and this one couldn't, now all of these can't. All of these can. All right. So even though if they had been pushed like this, now all of these can't and all these can't. All right. So how they arrange themselves in metaphase 1 along the metaphase plate can determine the expression of traits. And of course, you have those 4 sperm, right? And you don't really know which of those sperm is actually going to fertilize the egg. Did I talk about there are different types of sperm? Right? There are actually sperm that make like a defense, like they get close together and defend the ovum from any other sperm, so that their golden boy goes and does the fertilization. Okay, they like a defense. All right, and you know, a lot of mammals, they, you know, the female can be, there could be several donors, right? Just think of a dog or cat in heat, right? And all the cats around are following the cat in heat, right? All the males. And there are several donors, possible donors, right? But the one that inseminates first, you know, it would be best if they had some sort of way to stop other ones from fertilizing the egg once they fertilize it, right? So we have these defensive sperm in there, all right? It's wild, all right? But which of those sperm will actually be the one that fertilizes the egg? You know, we don't, you know, it's all random. So that could be, you know, one that can roll their tongue or one that can't roll their tongue. It could be either, right? Crossing over, then, remember, is where they, during prophase 1, where they swap actual arms of the chromosomes between the homologs. That is another way that you can get this genetic variation occurring. All right. So that is chapter 8. All right. Any questions? Any comments?
What do so in the two, like there are two different